Hello, I'm Dr. Knight at Cal Poly in San Luis Obispo in the Bailey College of Science and Mathematics. This is a series of lectures that we're doing for my evolution class, Bio 414, this summer, in summer 2025. We're calling it 10 lectures on evolution, and I'm doing this paper with my student.
Hi, I'm Keira Engler. I'm a biology and chemistry undergraduate student at Cal Poly, and we are working together on this presentation today. So this is the 15th target paper, and it was published in PNAS, and it's called Longitudinal Sequencing Reveals Polygenic and Epistatic Nature of Genomic Response to Selection. So we focused on some of the key scientists, Simon K.G. Forsberg, Lucas P. Henry, and Julian F. Areolis. These scientists have worked together for a really long time. This is another paper they published in Nature from 2021. So they've been working together, and they have a couple of other papers out together, but I just thought it was interesting how they're kind of a team. This one's about microbiota systems. They're microbiologists. And all three of them are now at Princeton University. And Lucas P. Henry got his postdoc at NYU. So we're going to start with by introducing some take-home points, which are things we want to focus on throughout this presentation. The first one being, evolution is more complicated than the textbooks say. You've been lied to. It often involves thousands of subtle genetic changes, each with a small effect on the phenotype. Replicate Drosophila populations were subjected to a high sugar diet for 100 generations, and allele frequency changes were studied. Thousands of genomes were sequenced at various time points. SNPs indicating alleles were tracked through time in control and high sugar treatments. They found that 4% of the genome was under positive selection. The selection response was very polygenic, meaning lots of factors. And few loci stood out as individual drivers of the gene for high sugar tolerance. Epistasis played an important role.
Back to Dr. Knight here. I wanted to read the abstract as we normally do, but in this case I've pasted in the abstract and I've shaded out the rest of the abstract. I just focused first on the first two sentences. This abstract was very information dense, and I think you can understand a lot about this paper just by understanding the abstract. So let's read this. Evolutionary adaptation to new environments likely results in a combination of selective sweeps and polygenic shifts, depending on the genetic architecture of traits under selection. I wanted to highlight these words, selective sweeps and polygenic shifts, and the genetic architecture. I'll go to those in the next slide. While selective sweeps have been widely studied, polygenic responses are thought to be more prevalent but remain challenging to quantify. Surprisingly, we know that this is happening, but it's until papers like this that we really see how it's working at the genomic level. So I'm going to do glossary notes as we go in the abstract. Selective sweeps are when a new beneficial mutation increases in frequency and becomes fixed within a population. This is often associated with reduced variability around the loci under selection. That's one of the ways we can identify those sites. Polygenic shifts are how multiple genes subtly influence a trait. In this study, polygenic shifts were studied across the timeline of the experiment. And then lastly, genomic architecture is a word that I've been stressing throughout the course. It's a more accurate and nuanced way of describing the genetic basis of a phenotypic trait. Now we'll go back to the abstract and introduce a few more sentences. So the first two sentences actually are kind of dense and not that important, but I'll read them anyway. The infinitesimal model makes explicit the hypothesis about the dynamics of changes in allele frequencies under selection, where only allelic effect sizes, frequencies, linkage, and gametic disequilibrium matter. It's a very dense sentence. And what I'm saying is that particular sentence doesn't matter that much. Next sentence, similarly, departures like this, like long-range correlations of allele frequency changes, could be a signal of epistasis in polygenic response. Epistasis is going to become and continue to be important to know about, but that sense is also pretty complex. We performed an an evolve and resequence experiment in Drosophila melanogaster, exposing flies to a high sugar diet for over 100 generations. We tracked allele frequency changes in greater than 1,000 individually sequenced flies in population pools and searched for loci under selection by identifying sites with allele frequency trajectories that differentiated selection regimes consistently across replicates. Still pretty dense, but I'm going to explain this, and their figures are pretty good. But let's first talk about what an evolve and resequencing experiment is. It's a type of study that investigates the genomic responses of adaptation during experimental evolution with tons of sequencing along the way. Identifying sites. These are sites that are basically SNPs that they identified after the fact, not beforehand like we did in the QTL studies. But these SNPs could be thought of as alleles that they differentiated, again, after the fact. Allele frequency trajectories is just another way of saying evolution. Smiley face, like allele frequency change in populations. That's our best definition of evolution. Back to the abstract. We estimate that at least 4% of the genome was under positive selection, indicating a highly polygenic response. The response was dominated by small, consistent allele frequency changes with few loci exhibiting large shifts. We then searched for signatures of selection and pairwise combinations of alleles in the new environment and found several strong signals of putative epistatic interactions across unlinked loci that were consistent across selected populations. Again, I'm sorry, these are long sentences with lots of dense information. Finally, we measured differentially expressed genes, or DEGs, across treatments and showed that DEGs are enriched for selected SNPs. Our results indicate, anyway, I messed up on the end of that. We're not going to have time to go back, but there was, just kind of ends, the abstract ends. But this word epistasis comes up. The effect of a gene or a mutation is dependent on the presence or absence of another gene or mutation. Keep that definition in mind. Remember, yesterday in lecture, I introduced the word epistasis along with pleiotropy. Very common words in evolutionary biology.
So another part of the paper we wanted to highlight was just this section. I'll read a couple lines. While early research focused heavily on identifying individual genes with large effects that undergo selective sweeps, we now understand that most adaptive responses involve more subtle changes across many loci. This emphasis on single gene effects has created a biased view of adaptation in the literature, even though cataloged examples exist in both humans and other species. Beyond identifying and just counting the number of genes involved, understanding adaptation requires considering how genes interact with each other, epistasis, and with the environment. So basically, this is kind of just saying, like, previous literature was wrong, and it's important that we know now that it's not the gene for hypothesis like we've talked about in this class. It's a bunch of different genes contributing in infinitesimal little ways. What? So one important glossary note in this paper that's come up and is important for understanding our results is positive selection, also known as directional selection. One extreme phenotype is favored over the extreme and moderate other types, causing a shift in allele frequencies toward the favored phenotype. So we're going to tie into our textbook about this, and the example they put is if you look at the graph on the y-axis, it's the number of individuals before selection, and on the x-axis, it's sort of just like a numbered average just for the example. And you can see that triangle sort of indicates that the median of the data, or the mean of the data, is right around 7, like 7 flat, and like right here. And after selection, we can see that that triangle is shifted to the right a little bit, indicating that a more extreme phenotype was favored, and thus the whole average shifted right up toward like almost 8. And this is a pretty subtle example, but you'll see in ours it's a little less subtle. And this is because the probability of surviving is a lot more favored at 14, with like a 1, the 1 being 100% survival versus the 0 being 0. And it's a positive association. So that's kind of why it pushed the average up toward the higher numbers. And this is just a different way of looking at this with like a bell curve. This is saying that like the distribution of like the peppered moth with like the light color, after selection, it got darker and the whole population shifted rightwards, which is just saying it survived better with a dark color because of the fires and like the soot-covered trees in its environment. And so the first figure in the paper, this is only one section of it. It was a pretty dense figure with a lot of great information, but I think the most important part of it was just this figure, which there's a lot of information at the bottom that we don't really need to get into. It's kind of talking about exactly how many numbers of flies they use, and being 100 or 200 across the 6 different control groups, which isn't super, we don't really need to get into that, but basically it's saying there's 3 populations of the control and 3 of the high sugar treatment based on the same NEX, which is like a type of fly mouse model, so like a base genetic model with no knockouts or anything. And it's just saying that some were for control, some are high sugar. So this is figure 2. I'm just going to show it all now, but I'm going to break it all apart. So if we look first at the legend, red is the high sugar group, black is the control group. And then we can see that for understanding each generation, they use different shapes. So like the very first generation, like your F1 cross, like it was a square versus like the 100th is a diamond. And one part that's really small in the figure is this is called a scree plot, and it basically is just mapping out a bunch of the PCs, and on the y-axis it says variance explained. So like if you look at this first bar, this is our PC1 in our PCA plot we're going to look at in the rest of figure 2, and it's just saying like 17% of this green bar, which is PC1, is 17% of the variance is explained, versus this is PC2, which is on the y-axis of figure 2, and it's saying around like 13.1% is explained, of variance is explained. So now we're going to zoom out, and this is the PCA plot. It looks a little different from ones we've seen in class because this is over the course of generations. So we can see the different shapes, like there's circles, diamonds, squares, and we can very much see like a divergence. And so when those points are getting farther apart, it's saying that they're more and more different. And we're basically looking at, from the 0 generation to the 100th generation, the control group had like a positive trend and the high sugar group had a negative trend with respect to PCA2. And it's basically just showing there was a large divergence, which we'll get into a little more in the other figures, but over time they got more different, which was pretty cool because our other PCA plots, it's just the grouping. This is the grouping and also on a time scale.
All right, back to Dr. Knight here. I'll try to explain figure 3, and I'm going to start with figure 3C. First, I want to explain the axes here. Generation, G1, G11, G25, G100. This is all 100 generations in this experiment. So we're going to look at data across these generations for allele frequency change. Remember, our definition of evolution is allele frequency change in populations through time. So allele frequency 0 to 1. We're going to look at 6 alleles, and 3 in the high sugar environment and 3 in the control environment. Those have different colors. And what I wanted to show first is an example where the high sugar environment had a significant effect on allele frequency change. Next, I'm going to show you all the data here. So you see the orangish tan bars are declining. That's the high sugar environment's having a negative response for these 3 alleles. And these are just 3 kind of random alleles that are under selection. Whereas in the control environment, those same alleles don't have any significant change through time. So just highlighting here a negative slope on the high sugar treatment for these alleles. Whereas in the control environment, it's a no slope, 0 slope. The slope will become very important as we go. Next, we'll look at where high sugar and control environments both had a negative slope for these 3 alleles. These are SNPs. They're SNPs that change through time. We can think of that as allele frequency changes through time. In both cases, there was a negative slope, negative for high sugar and also negative for the control. Next, also, there's some alleles that are only significantly affected by in the control environment. So there's evolution to the control environment, not just the high sugar environment. Here we see a positive slope for the control environment for those alleles and a no slope for the high sugar environment. Now we can look at all the SNPs at once. Remember, the SNPs are alleles. They're close to alleles that affect the phenotype. This is what's called a Manhattan plot. It's supposed to resemble the skyline of Manhattan. And this is slopes of all the SNPs. They basically identify SNPs after the fact. They got thousands of SNPs across the chromosomes of Drosophila, and you'll see bunches of different chromosomes here. Well, bunches of different SNPs, a whole bunch of different SNPs. First, let me explain the x-axis. 2L, 2R, 3L, 3R, X. Those are different chromosomes in Drosophila, but it doesn't make sense at first because wouldn't you think it starts with chromosome 1? But it doesn't, because chromosome 1 is usually the largest chromosome, but the sex chromosomes in Drosophila are fairly large. So this is a karyotype, a visual picture of the chromosomes in Drosophila. But we start at 2L. Now we see all the SNPs. This is the Manhattan plot. You see some points, each point is a different SNP. The slope of change through the generations is the y-axis. And that's the absolute value, whether it's positive or negative. And you can see the change.